uh, a continuation of your membrane potentials. So we have been learning about general physiology topics. We have learned about cell membrane junctions. We've learned about transport processes that happen. And in the last class, you have also learned about how potential difference exists in the cells at rest. Right? What are the mechanisms behind this? Okay. So today's class is about how potential changes when the cells are activated in some way, right? So let us understand why. Why should we know this topic first? So we are all aware of what happens in our day-to-day life, right? So we get this awareness from our receptors, sensations, all forms of general sensations, pain, touch, pressure, all of this. We can all feel all of it. We can also feel special senses. Vision, for example, hearing, all of this. So what is the basis that this stimuli which are outside the body, we are able to understand or we are able to be aware of it? How does it happen? Anyone? The stimuli which is present outside, in the surroundings, how do you become aware of it? What is the seat or biological site of awareness or consciousness? Yes? Receptors. So this is one part. Receptors are present on the skin. So what is the site of our consciousness or awareness? Skin is not the site of our awareness. Brain. So brain is what helps us to be conscious of everything, right? So receptor did some form of activity on the application of a stimuli, right? It could be mechanical, chemical, anything. And some way or the other, it was able to navigate its way to the brain, right? So brain tak kaise pahuncha wo? So in the form of some. So if you think of it, pressure sensation from your great toe, right? Your position sense from your great toe, this is one form of stimulus. So it is traveling all the way across your lower limb to the spinal cord and through that to the brain. So ये जो communication in the body, this happens is in the form of some electrical impulses. So impulse is the way in the form of which it travels. If I think of the opposite direction, so we perform movements on a day-to-day basis, simple movements like writing, right? Blinking, all of this. Any movement that you think of, it originates in the brain, conscious or voluntary movements. And from the brain again, through nerves, it is able to navigate its way to the effector organ. How does this happen again? So in some part of the brain, some activity arises in the form of impulses. It travels across nerves, different sets of nerves, and eventually it reaches the effector organ, in this case the muscles. So this brings us to the next one. So what is an impulse? If I ask you what is an impulse, not literally, but scientifically, what is an impulse? What would you call an impulse? With this word you will come across again and again. Impulses, in a very simple way it is. If you remember what you learned in the last class. Sudden change in. So impulses are waves of electrical changes, right? The long distance communication within the body happens in two ways. Either in the form of chemicals, hormones are the examples of that, and the faster one is the impulses of electrical potentials. So wherever you get this term, impulses are traveling from one part to the other, we are referring to electrical changes within the excitable tissues, which you call as action potential. So that is the topic for the day. So we'll define an action potential, we'll see what are the ionic processes involved in this, and we'll differentiate between a local potential and the action potentials. So till now what we already know from your previous classes is that there is a potential difference inside and outside of the cell, right? So we call this the resting membrane potential. So this potential difference is present in the rest, and some changes happen to this resting state during activity. But in activity also, it is not that equilibrium is reached. In activity again, the potential difference changes, right? So there also a difference exists. So that is, we call it as a action potential. So this we already know. Broadly, we use the word biopotentials to refer to this. So if you're asked about biopotentials, you talk about resting membrane potential, you talk about action potential, which is in frank activity, and local potential, which is somewhere in between. We have given some form of stimulation, but it's not of adequate strength. So there is some activity, but not as strong as an action potential. What is the difference? What is the reason of this variation in the concentration between the inside and outside of the cell? Which is the main ion inside the cell, if I ask you? The main ion inside the cell is potassium ion, right? And outside the cell, the main cation is sodium ion, correct? Very good. So what is the reason of this difference? So you have read about the Gibbs-Donnan effect, right? What does that say? Gibbs-Donnan effect. So in an ideal situation where we have diffusible ions on two sides of a semi-permeable membrane. This is an example. So both the solutions, solution A and solution B, they are electrically neutral, right? That means cations and anions on each side are balanced, right? Dono taraf. Also, the product of diffusible ions on this side is equal to the product of diffusible ions on either side. So this is a very ideal situation. But that does not exist in the body. So if we add one non-diffusible ion, so here we have added one non-diffusible anion on one side. So this leads to a variation of the concentration of the diffusible ions now, right? So here what we find is, right, the cation increases, right? The anion will decrease. So this presence of the non-diffusible ion changes the concentration of the diffusible ions. Why? Because eventually together they want to maintain the neutrality. So for this, the transport processes become active and it leads to this variation. So resting membrane potential का जो basis है कि inside and outside there is a variation of charged ions. The reason is the Gibbs-Donnan effect and the selective permeability of the cell membrane. Right? So inside the cell we have proteins which are non-diffusible, phosphates, sulfates. So these are all anions which are non-diffusible. Right? Okay, so this is the concentration. You've already told me about this. The differences. Now, sodium potassium ATPase, try to recall, this is a active pump, right, or active transport process. What does the active process mean? It requires ATP, and why does it require ATP? Because it is acting against the concentration gradient. So sodium is excess outside, in spite of that it is transported here by this potassium. Andar zyada hai, fir bhi potassium, it is pumped from outside to inside. So because it is acting against the gradient, it requires ATP. So this has got a role in all the cells. We'll come to this later on. From the preceding classes, we also know about ion channels, right? So what are these ion channels? Actually, you should try to recall what you learned in cell membrane. Cell membrane is a phospholipid bilayer, right? And ion channels are present or embedded into it at some intervals. So technically, what are these ion channels? These are proteins, right? So transmembrane proteins which can span the entire length of the cell membrane. So these are serving as conduits for ions, different ions to pass through, and hence the name ion channels. So these are important for very important for excitable cells. These are present in all cells, but specifically for excitable cells like nerves and muscles. So excitable tissue, when we call something as excitable tissue, it means they are able to generate and transmit the impulse over long distance. So that is what we call as a excitable tissue. But these channels, and for that matter sodium potassium pump, these are present in all cells of the body. Now ion channels are classified on various bases, like which ion are they letting in and out. This is the selectivity. sodium channel, potassium channel, because of their ion selectivity. So this is one basis of classifying it. Conductance, again, fast and slow on the basis of this. And mechanism of channel gating, right? What does this mean, channel gating? So these channels, some channels are present in their open state almost invariably without any alteration. You have read about leaky channels. So you'll come across this term, leaky channels. जो कि हर सेल में present है, leaky cation channels, which allow potassium ions from inside to move out, right, down their concentration gradient. So these leaky channels are open almost in their entirety. But mostly, if you leave around this set, all channels, they have an open and a closed state, and the mechanism by which they open and close, this is what we refer to as gating. So voltage gated channels, the one you see here, these channels open when there is a change in the voltage across the membrane, right? So resting state में inside is electronegative RMP, outside is positive. If there is any form of stimulation, this will change. And voltage change अगर है, that means if you are giving some form of electrical stimulation, these channels will open. So hence the name voltage gated channel. Second one is a ligand gated channel. So if a suitable ligand or a chemical binds, these channels will open. So one example of that is the acetylcholine gated sodium channels in the neuromuscular junction. So when acetylcholine binds to it, these channels open, letting in sodium ions to enter. And the third one that you come across, like example I remember right now, is in blood vessels. These are mechanically gated. That means mechanical stimulation, say, if there is a stretch of the tissue or the membrane, these channels will open. So this is how we refer to as gating. So these are very basic terminologies. You'll come across these channels throughout the year, right? But you should know what does this mean. So this is what voltage gating, ligand gating, and mechanical sensitive or stretch sensitive means. So I'll draw your attention to two channels that will come in context of today's discussion: sodium and the potassium channel. This is what a channel looks like: a transmembrane protein spanning the entire cell membrane. Now, you come across, you see that there are two gates, one activation gate, one inactivation gate. So, on either surfaces, one oriented slightly towards the outer aspect and one to the inner aspect. Activation gate and inactivation gate. So, these gates actually refer to the orientation or configuration of charged amino acid residues which are present inside. Right? So, because of the orientation of this amino acid chains, they behave like gates. Now, resting state में, right, when the cell is at say minus 90 millivolts or minus 70 millivolts, when the cell is at rest. So this is the orientation of the sodium channel. So it is closed from outside. So this is the activation gate. The inner one, the inactivation gate, is open. Right? Remember this. So when we have stimulated it somehow. So what happens is the activation gate now opens, right? And in this state or the activated state, both the gates, right, both these parts are open. So sodium ions can rush inside because of this state. And what you find here is the inactivated state. So as soon as the potential reaches to around plus 35 millivolt from the resting state, the inactivation gate closes, right? So it becomes closed from inside. So no matter what, kitna bhi sodium ion bahar present ho, if the door is closed from inside, it cannot come in. Right? Till the time it goes back to its resting state again. The sodium ion is unique in the sense that there is just not a resting and the activated state. We have an inactivated state in between. Right? Potassium ion, there is no inactivated state, so there is one resting state and one activated state. Resting state mein, the gate is closed, and when it's activated, the gate opens, right? But the difference in the nature is sodium channels inherently they are very fast, except for the nodal tissue of the heart, right? SA node, AV node, ये जो जगहें हैं, we refer to them as the nodal tissue. of the heart, except for these areas. Everywhere sodium channels are fast. They open fast and they reach their inactivated state within milliseconds. Potassium channels, by default, they are very slow. They open slowly and they close slowly. Okay. So resting membrane potential is negative, which means inside is negative compared to the outside. What are the factors responsible for this? This is a recapitulation. So the first factor is it is freely permeable to potassium and impermeable to sodium. Right? So what is the reason? We are not talking of activity now. We are talking of rest. As it is just there in the body like that. Why is it that these leaky channels are allowing potassium to pass and not sodium? If these leaky channels allow potassium to pass, the potassium will always travel passively from inside the cell to outside, and hence the development of negativity inside the cell. Right? What is the reason for this? Yes. Potassium is smaller than sodium. Who agrees with this? Do you try to recall the ionic structure of this Rutherford model that you studied? The nucleus, there are orbits, like. So sodium and potassium, which one is the larger ion? What is the atomic number of sodium and potassium? So which one is larger? Potassium is larger, right? Then how does this make sense? Leaky channels are usually not very selective, non-specific majorly. But in spite of that, body they have some form of selectivity. They will allow potassium but not sodium. Any guesses? Did this question come to your mind? The ionic size of the potassium is more, sodium it is less. But it is not allowing the larger ion to pass. Yes, because of charges. So what happens because of the charges? Orbits. Yes. Louder. So outermost orbit which one will have a greater force of attraction in the ionic state? Sodium ion, because the size is smaller, so on the outermost orbit they will have greater amount of attraction compared to potassium. So in the body, these ions exist in their hydrated state. So they are all bound to water molecules. So hydrated ionic size, when the ion is associated with the water molecules, the hydrated size of sodium ion is larger because it is able to attract more compared to potassium. So that is the reason the sodium ion in the hydrated state is larger. So that's why it cannot travel through it. Fine? So this is one way of looking at it. There are some other schools of thought which tell us that hydration ho ya na ho, that does not make a difference. When the ion is traveling through a channel, it just travels as the ion itself. So they explain the same thing on the basis of the energy required to strip the hydrated ion from the water molecules. The sodium ion में ज़्यादा water molecules associated है, so the cell has to expend more energy, right, to strip it of its water molecules and let only the ion in. So that makes the process difficult, right? So that is the reason of the first two things. Either of the ways you explain, but you should have an understanding. Right? It is impermeable to most of the anions because they are very large in size, and of course the activity of the sodium-potassium pump. Now when we have all the cells together, all the ions together in a cell, we can calculate the resting membrane potential with the help of one equation, the GHK equation, Goldman, Hodgkin and Katz's equation, right? Okay. So what is the role of sodium-potassium pump in the RMP? So sodium-potassium pump, if we talk of the genesis of RMP, कि -70 millivolt RMP है, तो उसमें -3 to 4 millivolt will be the contribution of sodium potassium pump. So you remember, that is why I have actually put it at the last. Because the contribution of sodium potassium pump in the production of RMP is less, very less. But it has got a major role in maintaining it. Once the RMP is attained, it is this pump which is maintaining it. Activity के बाद RMP जब restore हो रहा है, this is again maintaining the RMP. Now in nerves, skeletal muscles and all, this is all true, -3 to 4 millivolts. But in smooth muscles, the contribution of sodium potassium pump is higher. So smooth muscles may around -20 millivolts of the RMP is contributed by sodium potassium pump, right? So this is where it is an exception. Okay. Common terms that we look forward to today: depolarization, repolarization, and hyperpolarization. What does all of this mean? So RMP, we have been talking about that inside it is negative, outside it is positive. So this actually means the cell is in a polarized state. The rest में there is a definite polarity, inside negative, outside positive. Activity के साथ when this changes, we call this depolarization. That means the polarized state becomes, it is disturbed. So hence the name depolarization. So from negative, membrane potential starts becoming positive, right? And धीरे-धीरे it moves towards zero. And in some cases, it will even exceed zero, right? This is the process of depolarization. Now, from that depolarized state, when it is coming back to its original polarity, we call this process repolarization, right? So, depolarized state, say when the membrane potential becomes negative and it starts moving towards RMP, this is repolarization. And what is the other one? Membrane potential, say it becomes even more negative. So this is a hyperpolarized state. Jitna polarity hai, it becomes even stronger. So -70, say if it becomes -100 millivolt, this is a hyperpolarized state, right? Now let us start with local potential. So what is a local potential? तो हमने एक कुछ stimulation दिया उसमें, but it is not a very strong one. So some changes or disturbances will be there in the ionic distribution across the membrane, but these are not as strong as to produce an action potential. So this is what you call as a local potential or a graded potential. Okay. So remember when we talk of the ion channels, तो आपने देखा कि different gatings are there, mechanical, voltage, ligand, which actually means what? This implies that irrespective, right, of the nature of the stimulus, the effect is some ions will move in or out. This is what happens. Chemical ligand-gated में the stimulus is a chemical one, but जब वो channel खुला, so what will happen is some ions will come inside. Stress sensitive में भी वो है. So irrespective of the nature of the stimulus, the response is always opening of the channel. Okay. Now, depending on which electrode you are using to stimulate, so we have two forms or examples of this: cat electrotonus and anelectrotonus. So, cat electrotonus is seen when we are dealing with cathodal excitation, when you are stimulating the tissue with the cathode, right? And this is able to accept electrons. So, cathode accepts electrons, and hence the membrane where we have stimulated, the membrane under the electrode becomes positive, right? So cat electrotonus is hence a small depolarizing change. Suppose -70 millivolts से -63 या -55, तो यानी कि 7 से 15 millivolt का एक positive change develop हुआ. So that is a cat electrotonus, local change or a local potential, but it has not reached the threshold for that particular cell or nerve. When we are stimulating with the anode, so we call the potential that develops an anodal anelectrotonus, and because the anode, so correct this one, so because the anode is able to release electrons, the membrane under it becomes negative or hyperpolarized. So this is anode. So this is a mistake here. So anode releases or donates electron, leading to hyperpolarization. So when we have both the electrodes, this is what happens. The charges separate when the electrodes are present, and this type of potential differences which develop inside the tissue when electric field is set up, this is called as a electrotonic potential, right? And moving on to this local potential, so if you just now recall what you just learned, cat electrotonus is a depolarizing change. So minus 70, say, it will keep on increasing. If you give a small cathodal excitation, the response will be small. If you give a larger stimulation or excitation with the cathode, the response or change in the tissue will be larger. And if you increase it further, the response will even be more. Same exists for this. Anode, say, if I stimulate it slightly, we get a slight negative or hyperpolarizing change. If I stimulate it more, we get a more change, and likewise. So, here what you observe is the response of the tissue, or in terms of the local potential, depends on the strength of the stimulus. Jitna हमने strength दिया, the response is directly proportional to that strength of the tissue. So this is an important property of local potential, which is not true for action potential. So action potential में it is an all or none response. It is not this gradation. So that is why local potentials are also called as graded potentials, because there is a gradation. जितना हम लोग stimulus देंगे, magnitude of response will follow that. Now, cat electrotonus में, if it keeps on increasing, if the strength of the current that I'm using to stimulate, if I increase it, the response will keep on increasing. At some point, the response will just be enough to touch the threshold or the firing level for that particular tissue. So then what happens is, once it touches minus 55 millivolt, it will trigger action potential, right? So from here, what you understand is local responses, both the things, the cat electrotonus and electrotonus, both of them cannot produce an action potential. Why? Because action potential is a depolarizing change. So it is only when cathodal stimulation is given, action potential can be produced, right? तभी वो जाके positive की तरफ जाएगा, depolarize होगा. So an electrotonus cannot lead to action potential. Also, what we understand from this is local responses happen when the strength of the stimulus is lesser than threshold, right? तभी जाके we get local responses. So this magnitude of change, apart from the strength of the stimulus, it decreases in two things. The graph में जो आपको दिख रहा है कि with time, the response gradually dies down, right? So this is a decrement with time, and also if you know the stimulation से अगर आप जितना distance पे देखोगे, the magnitude again reduces with distance. Why? Because these potential changes develop because of reorganization of the charges. So there is a certain limit to which these charges will flow. So that is why they reduce with distance, and of course they also reduce with time. So this reduction, ये जो phenomena है, this is called as decremental conduction. So decremental conduction in terms of time and as well as distance. Now action potential. So what is an action potential now? So you define it by the sequence of events occurring in the membrane after application of a threshold stimulus followed by its restoration to the resting state. So I've underlined two words here. Why threshold? Because you've just seen in the discussion now, अगर वो threshold या adequate strength का नहीं हुआ, so it will produce some form of local change, some distribution or variation in the distribution of charges around the point of stimulation, right? And that will die off with time and distance. So that is not an action potential, right? So hence, in the definition itself, we should specify that sequence of events happening after threshold stimulus. Second point, word that I've underlined here is restoration. So it is just not the change that has happened, but also the coming back to its polarized state. ये पूरा sequence we call it as action potential. And these are the signals that I told you at the beginning of the class, which can travel along long distances without reduction in amplitude. So anywhere from the great toe to the brain or the opposite direction, if you want to move your toe or the ankle. So long distance communication needs कि ब्रेन से वो जैसे चला था एफेक्टेड ऑर्गन में वो वैसे ही पहुंचे, सो विदाउट एनी चेंज और रिडक्शन इन द एम्पलीट्यूड. सो दीज आर द पोटेंशियल्स व्हिच कैन ट्रैवल विदाउट रिडक्शन इन देयर एम्पलीट्यूड. सो एक्शन पोटेंशियल्स. If you look at the diagram, so what are the phases? So when you stimulate, so there is this initial phase. So suppose you start stimulation, initial response is slightly slow. So here the change that is happening is in the range of a local potential. But as it touches the threshold, there is a depolarized phase or depolarization. So from -70, it goes on to 0 and it overshoots to +30 millivolts even. So this phase is depolarization. From depolarized state, now again it comes back to the resting state. So this is repolarization. This particular phase, right, this is called as after-hyperpolarization, right? So even after the resting state has been achieved, what you can see here, कि कुछ देर तक वो और negative हो रहा है, and it continues for some time. The negativity goes beyond the RMP, hence the name after-hyperpolarization, and eventually after some time this comes back to its resting condition, resting potential. So all this is happening within milliseconds, right? So these are the phases of an action potential. Okay. Now what is the ionic basis? What are the events which happen during this process? So let us start from the beginning. So resting state mein, try to recall, potassium channels are closed, sodium channels are also closed. This is what is happening at rest. Now you have given a threshold stimulus. So a threshold stimulus is a form of electrical stimulus which opens the voltage-gated channels which are present in the membrane. Sodium channels, because they are very fast in their inherent nature, they immediately open. The sodium channels, when they open, they will allow sodium ions to pass. Remember, resting state में बाहर का दरवाजा बंद था, the door inside was open, inactivation gate was open. Sodium ion is a positive ion, right? So -70 millivolts से positive charges accumulate, positive ions accumulate, and it takes the potential towards the positive side, 0 and even towards +30-35 millivolt तक उसको ले जाएगा. One more event happens here. I'm coming to this in a bit. But once it reaches +30 to 35 millivolts, now cell has become positive, now electropositive, right? Now for the sodium ion, the electrical gradient has now reversed. Rest में कैसा था कि अंदर नेगेटिव था, बाहर पॉजिटिव था. So there is a natural tendency of a positive ion to come towards a negative place. Electrical gradient for sodium was from outside to inside. Now when it has reached +30 to 35 millivolt, that means उस point पे the polarity has reversed. Inside is now positive and outside is negative. Polarity has reversed. So now there is no gradient, electrical gradient for the sodium ions. The sodium ions stop moving in. Sodium ions also activated state say they enter the inactivated state. Remember inactivated state वो state था जिसमें अंदर का दरवाजा बंद है, but it's open from the outside. But once it is closed inside, no matter how many charges, even if they are present, they will not be able to navigate in, right? And at the same time, potassium channels start opening. तो वो भी voltage gated potassium channels ही हैं, they start opening. So for potassium ions, the gradient is from inside to outside, right? In both ways. Electrical gradient भी reverse हो चुका है, so a positive ion will try to move out, and chemical gradient तो था ही, chemical gradient potassium अंदर ज़्यादा है, so it will move out. So with this potassium ions, this potassium channels opening, potassium ions move out, right? So again, when a positive charge moves out, दोबारा से वो negative की तरफ जाना शुरू करता है, and at some point it will come back to the RMP. But potassium ions, in their basic nature, they are slow. So even after they reach RMP, they have not yet closed. तो बंद हो रहा है, but they have not yet completely closed. So some potassium efflux still continues, right? So this takes it to a bit more negative state. So minus 70 को cross करके it may reach to minus 80 or minus 85 millivolt. This is the phase of after hyperpolarization, from where it again comes back to its resting state as the potassium channels close and the sodium potassium pump comes into the picture to bring it back to its resting ionic composition. So these are the phases and the events that happen in the different phases of action potential. So two things at least that you should remember is depolarization पे there is sodium influx and in repolarization there is potassium efflux. These are the two keywords that you remember. So initially sodium channels are full, the initial response is slow, but as soon as they reach the firing level वो level है जिसमें membrane potential में कुछ ऐसा change हुआ, the voltage change that has happened is of such an extent that all the voltage gated sodium channels in that part they open simultaneously, right? Why? Because remember channels में हमने जब बात किया था voltage gated channels open when there is a change of voltage. So initially when you stimulate, some channels open. From these channels some sodium influx happens and positivity starts developing, voltage change starts developing. As soon as a certain extent is reached, we call this the firing level or the threshold level, all the voltage gated sodium channels will open, or sodium ions will come inside, or voltage change hoga, or sodium voltage gated channels will open. So this is a positive feedback cycle which develops here. This is called as the Hodgkin cycle. So this is responsible for this sharp rise in the depolarization. Otherwise it would have risen slowly, but here we find the spike. So that is the reason. So these are the two events that happen in the depolarization phase. Repolarization, as I said, there are three things that happen. Sodium channels enter the inactivated state, right? And they're not in their resting state, remember. Resting वो बाद में आएगा, but they are activated के बाद inactivated state. दूसरा, I just told you direction of the electrical gradient. This reverses, right? So that's why sodium influx stops. This is one more reason. And the third is opening of voltage-gated potassium channels. So these are the two things. After hyperpolarization, if I ask you what is the cause, this is because of the basic nature of the potassium channels that they are slow to begin and slow to close. That's why even after achieving RMP, channels take some time to close. उस time में कुछ potassium as they enter, the efflux continues and it becomes more negative. And RMP is attained by the activation of sodium potassium pump. Okay. Now, how many of you remember this story? All of you have read it in your school, right? So I find a very nice analogy here that the rabbit was fast to start, right? And then after, because it was very fast, it was at that speed, so it slept and dozed off after some time, right? So this is how a sodium channel behaves. It is very fast to begin with, and within the fraction of milliseconds, it again goes to the inactivated stage. This is the nature of the sodium channel. And the tortoise is the potassium channel. वो धीरे धीरे चलता है. So खुलता भी है धीरे धीरे and it stops also gradually. So this is actually what happens in the channel conductance of both the ions. And why is it like that? कि दोनों ही channels voltage gated है. Sodium हो या potassium हो, दोनों voltage gated है. But दोनों action potential अलग अलग phases में खुल रहे हैं. What is the reason behind this? But the reason is the nature of the channels. We use the word conductance to refer to this ease of opening and closing. So it is the same depolarizing stimulus. तो हमने जिस stimulus से दिया था, it is the same stimulus which opens both the sodium and the potassium channel. right? But sodium channels, because they are fast, so they coincide with the depolarization phase. You give a fast end. And potassium channels, धीरे धीरे वो खुले हैं तब तक, right? So opening of the potassium channel coincides with the repolarization phase. But remember, it is not something other stimulus which is coming. वही stimulus जो हमने दिया था, the same stimulus is causing activity of both the channels. But such is the beauty that it coincides with two phases of the action potential. Okay. So properties will take up in the next class. So after discussion में, can you tell me one or two properties that you could understand? The discussion today. Local or action potential में, what was that one word that was very the hallmark of both these two? For local potential, the stimulation has to be sub-threshold. For action potential, it has to be threshold, right? So that means one of the important properties of action potential should be the threshold intensity. If we are not giving a threshold intensity stimulus, we will not get a response, right? So this is one. Also recall, the local potential ka graph tha. So there was some form of gradation, right? Tissue mein, I'll just take you back a few slides. Yes. So the magnitude of the response depends on the strength of the stimulus used. But same is not true for an action potential, right? So gradation is possible. It is present in a local potential. Hence, it is also called as a graded potential, and it is absent in an action potential. Action potentials follow a all or none law. What is that? If you give a stimulus, if you give a threshold stimulus, response will be maximum, all response. And if it is sub-threshold, there is no response. You may get some changes, but you will not have the depolarization, repolarization, the proper phase. So there is none response. If you increase it beyond threshold, tab bhi response is already maximum. It cannot increase further. So action potential does not follow gradation, it follows the all or none response. So these are the two things I think you can understand from the discussion today. Achha. If you give two stimulus, for example, two stimuli, can these two stimuli add up and bring a change in the response? Think of it. Action potential, if you talk of action potential, if you give two stimuli in quick succession, can they add up and produce a response? Or can they add up or increase the response? No, right? And in this, local potentials is it possible? In quick succession, if you give two stimuli before the first one decays, so can it increase the response, magnitude of the response? It can, right? Because यहाँ पे gradation हो रहा है, it has not reached its maximum. So this property is also called as summation. Summation literally भी उसका meaning वैसा ही है, addition. So when we give more than one stimuli at multiple points at one time or in quick succession, एक ही जगह पे बार-बार, action potential does not show summation, but graded potential, local potentials can show summation. Right? We'll stop here. Next class we'll talk about the properties of action potential and the applied aspects. All right. Thank you.